this photo, I am playing the role of a pesticide-resistant bacteria in order to provide evidence for evolution. Evolution is the change in characteristics of species over several generations and relies on natural selection, or simply a change in genetic frequencies over time. With this comes evidence to prove that evolution is real. One of those evidences would be observed evolution, and an example that comes from that is pesticide-resistant bacteria. Over time, some bacteria of a population becomes resistant to the pesticide. The survivors reproduce, and the resistance becomes inherited, increasing the number of resistant bacteria. In this photo, I am standing on one of the two islands to represent the works of allopatric speciation. Allopatric speciation is a geographic isolation where the same species separate from each other. This action prevents or interferes with gene flow. The end result of allopatric speciation causes origination of new species. This ultimately leads to evolution because the splitting of one species to a different area that has a barrier between them increases the original species into many different ones. In this photo, I am pulling in the mitochondria and the chloroplast to form the endosymbiotic theory. The endosymbiotic theory is an evolutionary theory that assumes that eukaryotic cells came from prokaryotic organisms. It is said that mitochondria came from aerobic prokaryotes and chloroplasts came from cyanobacteria. These two organelles were once individual organisms 1.5 billion years ago, and the evidence to support that is that they reproduce independently, produce their own energy, have their own DNA, have their own ribosomes, and are double-membraned. In this photo, I am slicing the bread to better understand the components of inside a carbohydrate. Carbohydrates are composed of CHO, which is carbon, hydrogen, and oxygen. It functions as the main source of energy for the body. Also as energy storage, building materials, and raw materials. They have a monomer called a monosaccharide, which is a single sugar, which soon forms a polymer called a polysaccharide. Carbohydrates have glycosidic linkages that joins carbohydrate molecules to another group. Examples of this important biomolecule includes potatoes and bread. In this photo, I am knocking on the cell membrane to ask for permission to enter since it only allows some things to pass through. The cell membrane is selectively permeable, meaning it only allows certain materials to pass through the membrane. This is very important to help the cell maintain homeostasis. Also, with the semi-permeable structure, nutrients are able to enter the cell and wastes are able to leave the cell. Another name for the cell membrane is called the fluid mosaic model because it is a flexible bilayer of phospholipids embedded with proteins and other molecules. In this photo, I am representing an enzyme that has been denatured due to salinity. Enzymes are macromolecules that are biological catalysts. They have factors that affect their ability to function correctly. An example of a factor affecting enzymes is salinity. If the level of salinity is beyond or below the optimal level, the enzyme will denature, which means it will no longer function as it used to, becoming useless. Factors affecting enzymes are important to learn and understand to properly maintain the original functions of the enzymes. And this concludes my project. Thank you.